When highly acidic chyme empties from the stomach into the duodenum, it presents an immediate caustic threat to the delicate intestinal mucosa. This creates a secondary problem. The complex macronutrients suspended within this acid, the fats, proteins, and carbohydrates, cannot be absorbed in their current state. Survival requires the gastrointestinal tract to simultaneously neutralize this acid and chemically dismantle the macromolecules. This diagram shows the primary physiological countermeasure, the exocrine pancreas, nestled into the duodenum. The main pancreatic duct, the duct of Wirsung, acts as the central fluid highway. At the terminal intersection, it joins the bile duct, entering the intestine via the sphincter of Oddi. The fluid entering the duodenum through this sphincter functions as a highly coordinated dual-component system. There is an egbolic component, the dense enzymatic payload responsible for digestion, and a hydrolytic component, the aqueous bicarbonate flush responsible for pH neutralization. To understand gastrointestinal survival, we must conceptually split the pancreas in two, examining the distinct microscopic engines that drive these separate fluids. This histological cross-section reveals the source of the enzymatic payload. The vast majority of the tissue consists of lobular clusters of sacs called acini, marked here as number 1. Zooming in on a single serous acinus, we see a single layer of pink pyramidical epithelial cells forming a central lumen. Their primary function is packaging digestive enzymes, stored as stippled zymogen granules, densely packed near the inner apical poles. These enzymes, such as trypsinogen, are packaged in completely inactive forms, alongside trypsin inhibitors, to prevent the pancreas from digesting its own tissue. Releasing this payload requires specific neuroendocrine triggers, cholecystokinin, or CCK, released from intestinal eye cells, and acetylcholine, delivered via parasympathetic vagal nerves. This schematic of an acinar cell shows how these triggers operate. At the basolateral membrane at the top, specific G-protein-coupled receptors bind the acetylcholine and CCK. Tracking the signaling cascade downward, both pathways funnel into an explosive reaction, a massive spike in intracellular calcium. Following the final arrow, this calcium spike drives exocytosis of zymogen granules, dumping enzymes into the lumen. This resulting fluid is the primary secretion. It is an incredibly dense, low-volume sludge packed with latent chemical potential. The acinar engine successfully arms the chemical weapons needed for digestion. However, this concentrated enzymatic sludge is completely immobile and useless without a secondary delivery system. Looking at this diagram, we can follow that dense primary secretion as it is pushed out of the acinar lumen and into the connecting transit system. The columnar epithelial cells lining these ducts are active secretory engines, specifically exchanging chloride and bicarbonate across their membranes. The regulatory trigger for this ductal engine is entirely different. It requires secretin, a hormone released by duodenal S cells, strictly in response to high luminal acidity. Unlike the acinar cells that run on calcium, secretin activates these ductal cells by elevating intracellular cyclic AMP. This schematic inside a duct epithelial cell maps the biochemical machinery. On the left, carbon dioxide and water combine via the enzyme carbonic anhydrase to form carbonic acid. This carbonic acid immediately dissociates, splitting rightward into a hydrogen ion and a bicarbonate ion. The hydrogen is ejected back into the blood in exchange for sodium. The remaining bicarbonate must now be moved into the duct. Cyclic AMP physically opens a specialized gateway on the luminal membrane, the CFTR channel, dumping chloride into the duct lumen. This expelled chloride establishes an elegant ion exchange loop. A neighboring transport protein pulls chloride back into the cell to pump stockpiled bicarbonate into the pancreatic juice. The massive efflux of bicarbonate into the lumen creates a strong osmotic gradient, pulling sodium and copious amounts of water across the membrane with it. This creates a high-volume, highly alkaline, isotonic flush that dilutes the acinar enzymes and transports them forward. This ductal bicarbonate flush is the singular mechanism capable of raising duodenal pH to 8.0. This alkalinity provides the specific environment required for pancreatic enzymes to achieve their optimum catalytic activity once they reach the gut. Chronologically, the nervous system anticipates the need for this pancreatic fluid long before food actually reaches the intestine. Looking at this temporal sequence, the top right details the cephalic phase. The mere sight, smell, or taste of food sends feed-forward cholinergic signals via the vagus nerve directly to the acini. Moving to the gastric phase on the left, physical distension of the stomach reinforces this reflex and stimulates the release of gastrin. Tracing these pathways to the center, they trigger an enzyme-rich pancreatic secretion, accounting for roughly 30% of the total volume. The dynamic shifts rapidly during the intestinal phase, beginning the moment acidic, fatty chyme breaches the pyloric sphincter and enters the duodenum. On the bottom left, the sudden drop in pH triggers S cells to dump secretin, while fatty acids trigger I cells to release CCK. Following the path down, this accounts for the remaining 60% of total secretion, unleashing the massive, high-volume aqueous bicarbonate flush. The brain efficiently preloads the enzymatic artillery while the food is still in the stomach, allowing the intestine to simply pull the trigger on the neutralizing flood at the exact second of impact. The lethal potential of the pancreatic payload is fully realized when the precision of this dual-engine system breaks down. Consider a failure of the ductal engine. If a genetic mutation alters the CFTR chloride channel, the cyclic AMP pathway is severed. Without chloride transport, the osmotic gradient collapses. Water fails to flow, turning the primary secretion into a stagnant, viscid sludge. This pathophysiology is the hallmark of cystic fibrosis, leading to devastating mechanical obstruction and severe malabsorption. Conversely, consider a failure of the acinar safety mechanisms. We have to ask what happens if the intracellular trypsin inhibitors fail to suppress the zymogens. In this catastrophic scenario, trypsinogen prematurely converts into active trypsin while still packaged inside the pancreas. Because active trypsin cleaves and activates the other stored enzymes, this single error triggers a runaway proteolytic cascade, turning the gland's weapons against its native tissue. This rapid, excruciating state of autodigestion is acute pancreatitis. Exocrine pancreatic function is a masterpiece of controlled volatility. It relies on keeping the enzymes inactive, perfectly separating the calcium and cyclic AMP intracellular triggers, and timing the bicarbonate flush with exact precision. Human digestion relies on the flawless, phase-locked synergy between the acinar payload and ductal delivery system.